Hi, hello, welcome to my channel, Dr. Srimani Sesham, presenting to you another important topic in genetics crossing over. If you have not yet subscribed to my channel, please like, share, and subscribe, and hit the bell icon for further notifications. Now, let us get into the topic crossing over. Crossing over may be defined as the recombination of linked genes brought about as a result of interchange of corresponding segments between the non-sister chromatids of a homologous pair of chromosomes so as to produce new combinations of genes. The chromatins resulting from such interchanges of chromosomal parts are known as crossovers. Now let us see what are homologous chromosomes. A pair of homologous chromosomes are a set of one maternal and one paternal chromosome that pair up with each other inside a cell during fertilization. The chromosomes in a homologous pair each have the same genes in the same order, but there may be a variation between them resulting in different alleles. As we can see here in the diagrammatic representation, the two homologous chromosomes are represented in two different colors, so that you can see that one of the chromosome is green and the other one is red in color. So when there is a crossing over taking place between two such homologous chromosomes, the arms of each one are crossing here, which are called as the non-sister chromatids. Okay, so the crossing over takes place between the non-sister chromatids, where exchange of the DNA takes place between them. So what are sister chromatids? Sister chromatids refer to the identical copies formed by the DNA replication of a chromosome with both copies joined together by a common centromere. So here we can see the sister chromatids in the diagram, where you can see they are joined by a common centromere, and these are called as the homologous chromosomes, and these are the sister chromatids. Whereas when there are two such homologous chromosomes and the crossing over happens between these two, then the arms, here you can see these are the arms of two non-sister chromatids. Otherwise, this is one sister chromatid and this is other sister chromatid. So non-sister chromatids refer to either of the two chromatids of a pair of homologous chromosome, that is the pairing of a paternal chromosome and a maternal chromosome. So coming to the types of crossing over, crossing over is of two types basing on their occurrence in somatic or germinal cells. So in case of somatic cells, if it is occurring, it is also referred to as mitotic crossing over. So somatic crossing over was first reported in the somatic cells of Drosophila by Kurt Stern, and when the process of crossing over occurs in the chromosomes of body, the autosomes or somatic cells of an organism during the mitotic cell division, it is known as somatic or mitotic crossing over. The somatic crossing over is rare in its occurrence and has no genetical significance, because if it is occurring in the autosomes, okay, it is not inherited, while if it is occurring in the allosomes or the sex chromosomes, then definitely they will be inherited. That is why they have the genetical significance. Now coming to the second type, germinal or meiotic crossing over. So usually crossing over occurs in the germinal cells during the gametogenesis, in which meiotic cell division takes place. So during meiosis, we can see the occurrence of crossing over. This type of crossing over is known as germinal or meiotic crossing over, and it is universal in its occurrence and is of great genetic significance. As I've already told you, it is of great genetic significance because the germinal cells are the ones which are inherited. Okay, the characters or whatever exchange occurs, the change or the variations that occur are inherited to the offsprings. Now let us see the major steps that are part of the crossing over mechanism. So the first step in the mechanism of crossing over is synapses. So during prophase 1 of meiosis, the maternal and the paternal chromosomes of a homologous pair come close together and pair at synaptotene or zygotene substage. So this pairing is remarkably precise and point to point. The homologous chromosomes come close together at one or more points by mutual attraction between the allelic genes. At the end of zygotene, they lie side by side in close approximation all along their lengths. The pairing is exact and the paired chromosomes are known as bivalents. The pairing phenomenon of homologous chromosomes takes place in the presence of synaptonemal complex. Here we can see how the pairing is happening between two homologous chromosomes and in the presence of the synaptonemal complex. The synaptonemal complex contains DNA and protein. So that marks the first step in the mechanism of crossing over. Coming to the second step in the mechanism of crossing over, that is the duplication of the chromosome. During diplotin stage, each of the homologous chromosome in a bivalent splits longitudinally into two sister chromatids. Thus, the bivalent now consists of 4 chromatids and is known as a tetrad. The longitudinal splitting of chromosomes is achieved by the separation of already duplicated DNA molecules. The third step in the mechanism of crossing over is the exchange of chromosome and segments between two non-sister chromatids, that is the crossing over. During diplotin stage, when the paired chromosomes start separating, the chromatids remain in contact at one or more points and thus establish one or more exchanges per bivalents. These points of contact are known as chiasmata. At each chiasma, two non-sister chromatids of the bivalent break at the corresponding points and then rejoin with exchange of segments. The new chromatids formed as a result of exchange of segments are formed of segments derived from two non-sister chromatids of the bivalent here. So the next step will be terminalization, the fourth step. After crossing over, the non-sister chromatids start repelling each other because the forces of attraction keeping them together will lapse. The chromatids separate from the centromere towards the tip, and in so doing, the chiasmata also start moving in zipper-like fashion towards the end. So, the movement of chiasmata is known as terminalization. Simultaneously, the chromatids condense and get shortened, and in diakinesis, the homologous chromosomes become separated except at their ends. So, now let us see what are the enzymes that are involved in crossing over. So, the enzymes that are involved in crossing over are the recombinase, which is the major enzyme regulating the recombination event, the endonuclease, which is the enzyme responsible for breakage of two non-sister chromatids at corresponding sites, and a ligase enzyme, which is the one helps in the exchange of segments, and finally the exchange segments are joined or the gap is filled by the ligase enzyme. Now coming to the kinds of crossing over. The kinds of crossing over are a variable number of chiasmata which are formed. Accordingly, they have been classified. So there can be variable number of chiasmata along the length of a chromosome, and the number of chiasmata usually depends upon the length of the chromosome. Depending upon the number of chiasma, crossing over can be of the following types. The first one being the single crossing over, when only one chiasma is formed all along the length of a chromosome pair. It is known as single crossing over. The chromatids of chromosomes contact and break only at one point along their entire length, as seen in the depiction here. These are the two homologous chromosomes, and the crossing over is happening between the two chromosomes only at one point. Okay, after the separation or the breaking, you can see the exchange has already happened between these. So these are the new recombinants that are formed. So that is single crossing over. Now coming to double crossing over. Double crossing over chromatids break and rejoin at 2 points. It means two chiasmata are formed along the entire length of the chromosome. In double crossover, the formation of each chiasma is independent of the other. It means each crossover can involve either of the two chromatids of each homologous chromosome, so that three different types of crossover tetrads are formed. The first one being two-strand double crossover, which is also referred to as the reciprocal chiasmata. In this, the same two chromatids are involved at both the crossing over points. The chiasmata thus formed are known as reciprocal chiasmata. As a result of this, two chromatids are non-crossovers, and the presence of the parental combination of the genes is there. And the second type is three-strand double crossover. Three-strand double crossover, in this crossing over, three chromatids are involved. That is, the second chiasma contains one of the same chromatids which has crossed at first chiasma, but the other chromatid is different. Here only one chromatid is non-crossover, while the other three exchange their segments. And then the third one being four-strand double crossover, which is also referred to as the complementary chiasmata. In this type of crossing over, all four chromatids of the tetrad are involved. Two of them exchange parts of the chiasma, and the other two are involved in the second chiasma. These produce four single crossovers, but no non-crossovers are present, because all four have participated in the crossing over. Then the last type being multiple crossing over, that is, more than two chiasmata are formed in case of multiple crossing over. So when crossing over occurs at more than two places in the same chromosome pair and more than two chiasmata are formed, this type of crossing over is known as multiple crossing over. It is very rare because of interference in crossing over. Now let us see what are the factors that control the frequency of crossing over. Frequency of crossing over is dependent upon the distance between the linked genes, but a number of genetic, environmental, physiological factors also affect it. They are as follows. Let us see each one of them. First one being the temperature. High and low temperatures increase the frequency of crossing over. The second one being the X-rays. So the X-rays, Muller has discovered that exposure to X-ray and other radiations increases the frequency of crossing over. The third one being the age. The frequency of crossing over decreases with increasing age in female Drosophila. That was a study done on Drosophila, from which they have come to the conclusion about the controlling factor as age. And then the next one being chemicals. Certain chemicals which act as mutagens do affect the frequency of crossing over. Some increase the frequency, whereas some decrease the frequency of crossing over. The next one being the sex. So crossing over in males of Drosophila is negligible. In silk moth, crossing over does not occur in females. Like they have studied in certain insects where the sex has an influence on the occurrence of crossing over. Then the next factor being chiasmata formation. So the chiasmata formation at one point discourages chiasmata formation at other point in the vicinity. So this phenomenon is known as interference, which we have seen the multiple crossovers. At that time I had referred to the term interference. So that is this, the chiasmata formation, if it happens in one place, will not allow the formation of the chiasmata in the vicinity. That is called as interference in chiasmata. And then next one being inversions. Inversion of chromosome segment suppresses the crossing over. And then the distance between the linked genes is the major factor which controls the frequency of crossing over. The chances of crossing over between distantly placed genes, cytoplasm. So again, cytoplasm also has an effect on the factors which influence the frequency of crossing over. So factor of crossing over is present in the cytoplasm and is inherited to the offspring. Then the nutritional effect. Crossing over frequencies between the same loci in different strains of the same species shows variations because of numerous gene differences. So then the metallic ions such as calcium and magnesium, these are affected. The concentration of these ions also affects the frequency of crossing over. Then genotypic effect, as I've told you, the variations which occur are because of numerous gene differences, whether within the same species, the strains of the same species, or of the same loci. Okay, then we can see the genotypic effect also influencing the crossing over. Then chromosomal structure effect. So genes which are changes in the order of genes on a chromosome produced by the chromosomal aberrations usually act as crossing over suppressors. And then finally the centromere effect, where the genes present close to the central region show reduced crossing over here. So these are the various factors which control the frequency of crossing over. Then what are the vital features of crossing over? Some of them are as follows. Let us see them. Only 2 of the 4 chromatids cross over, while the other 2 chromatids preserve the original constitution. The crossing over occurs between the non-sister chromatids of the homologous pair of chromosomes. The 2 chromatids of the same chromosome never exchange segments. The number of chiasmata per set of chromosomes depends upon the length of the chromosomes. The longer the chromosomes, the greater the number of chiasmata. Chances of crossing over are more if the genes are located farther apart. The more is the distance between the genes, the greater is the opportunity of chiasma formation. As we have already seen, if the vicinity, that is the formation of chiasmata, is not in the closer vicinity, which is termed as the interference in case of crossing over. So let us now see the significance of crossing over. Crossing over provides a strong proof in favor of linear arrangement of genes on the chromosomes, and the recombinations or new gene combinations are produced due to crossing over, which change genetic pool by changing of gene frequency, and this provides a way of evolution. Crossing over generates genetic difference within a population. Linkage map and genetic maps are constructed on the basis of crossing over. So Thank you for watching my video. Please like, share and subscribe, and do hit the bell icon so that you will get further notifications.